So the title of my talk is a song of the Bee Gees, 1970. I've been warned by one of my students recently that I shouldn't use these talks because they tell my age, and which is probably true. But I think that it gives well the idea of what we try to do in the laboratory. So when I was a medical student in the 80s, there was an atlas of medicine, very famous one with anatomic tables. If you have studied medicine, some of you will come across on the drawings of Frank Netter, an MD from the United States, who produced these fantastic drawings and anatomic tables. And on the page of myocardial infarction, there is this drawing. So this is a relatively old person. It's winter outside, so it's cold. He's carrying a weight. He just came out of a restaurant, so he had a good meal. All these are aggravating factors. And he has pain in his chest because he has a myocardial infarction, so what's called in the Anglo-Saxon terms heart attack. And the reason why he has pain, because the heart attack is due to the closure of the coronary artery, so the portion of the heart below the coronary artery is not a sufficient supply of oxygen and energetics, and so he starts dying. And this patient can die immediately by arrhythmias, but he usually doesn't die because of the infarction itself. And so if he's resuscitated or brought rapidly to the hospital, then he has a very high chance to survive. However, the portion that is damaged of his heart tends to become bigger. And the only way that it can get healed is through formation of a scar. But a scar inside the myocardium is an impediment to good contraction, and it is a loss of muscle mass. And so he has a high probability to develop a condition in which the heart enlarges, doesn't pump sufficiently, and this is known as heart failure. Once a person has heart failure as a diagnosis, it's a 50% chance of not being alive after only 4 years. So it is a prognosis that is much worse than several cancers. If he's lucky and lives close to an hospital and the emergency system works well, then he's brought to a cath lab, so an intensive coronary unit. An interventional cardiologist inserts a catheter into his coronary artery, opens up a balloon. This is called percutaneous coronary intervention or angioplasty, and this reduces the lost portion of myocardium because it restores flow. However, what is dead, and then eventually is repaired by a scar. And the reason why there is scar formation, because our left ventricle is formed by approximately 10-12 billion cardiomyocytes, so contractile muscle cells, which are unable to replicate. You can take these cells out of the heart, you put them in culture. They are very big. These striations are the contractile apparatus that permits them, these cells, to contract. Sometimes they are binucleated, but you can do everything to these cells in terms of electrophysiology, molecular biology, immunofluorescence, all kinds of studies, but you cannot push so far these cells to proliferate. So basically, we are born with a certain number of cells in our heart, and these cells stop dividing immediately after birth, and we live for the rest of our life exactly with the same cells in the heart. The reason why the heart of a baby is this big, the reason our heart is this big, is simply because not the number of cells increase, but the size of the cells. And this is why they have these enormous cytoplasms. So you say, okay, so since this is a problem, we should inject these patients with a drug that protects his heart during acute damage until there is revascularization. And then, since we are ambitious, we want to inject a drug to these patients here that might induce his heart cells to proliferate and regenerate the heart. And this problem of losing cells in adult life is a key problem of medicine now, because if you look at the statistics, after 80 years of age, 1 person out of 3 has a dementia. In most cases, Alzheimer's disease. Alzheimer's disease is loss of cholinergic neurons in the brain. You lose cells, and the brain is not able to regenerate these cells. 1 person out of 2 after 75 years have loss of don't hear well, and this is because they lose neuroepithelial cells in the inner ear. So they lose these cells and don't regenerate them. 1 person out of 3 after 75 doesn't see well, because they have loss of cells in the retina, and the retina is incapable of regenerating. The way I teach to my medical student now is teach your patients to take care of their cells, because they are endowed with a certain number of cells at their birth, and if they take care of them, these cells will resist longer and they will live longer. If they don't take care of these, then the cells are gone, and then disease and death comes. You will say, well, there must be drugs for protecting the cells, for regenerating the cells. Well, there are none. There are none for the brain, for the retina, for the inner ear. There are none for the heart. There is nothing that can protect the heart. And there is nothing that can protect, that can induce the heart to regenerate. All the drugs that are available are drugs that try to convince the existing cells, or the survived muscle cells, to perform a little better. And most of these drugs have been, all of these drugs, except this one which is in clinical trial now, all these drugs have been developed, the last one in the mid-90s. So it is 20 years that in the best clinical setting, treatment of patients with myocardial infarction, with heart failure, is exactly with the same medications that would have been used 20 years ago. We are very intrigued by both cardioprotection and cardiac regeneration, and we are trying to develop drugs for both conditions. We speak about cardiac regeneration today, which is not a trivial problem, because when there is a myocardial infarction, so a heart attack, from 2 to 4 billion muscle cells are lost from the heart. And there is no understanding why these cells don't proliferate. Especially, there is no understanding why these cells stop proliferating at birth. Birth is a very dramatic event for a mammal, for a baby. There is a tremendous increase in mechanical stretch, because before it was the heart of the mother supporting most of the circulation. There is a lot of oxygen in the heart at the birth, because before birth, in the womb of the mother, the heart was a venous organ, very far from the lungs. Immediately after birth, it is flooded by oxygen from the lungs. A few years ago, together with Serena Zacchina here at the ICGEB, we collaborated in a study which showed that oxygen stops proliferation of cells, but it's not the whole story. This year, again, together with Serena, we found that there are maternal factors produced by immune cells of the mother, and in particular T regulatory cells that secrete factors that induce cardiac cell proliferation. And these T regulatory cells are very interesting, because the baby who is growing in the mother is genetically different 50% from the mother, so it must be tolerated. It's physiologically tolerated. It is tolerated because there is an expansion of this class of lymphocytes that induce immune tolerance, and some of these lymphocytes produce factors that induce cardiomyocyte proliferation. So that's a very intriguing story, but there is not a definitive answer on that. And since there is no answer in terms of mechanism, people have tried to supply exogenous cells to the heart to replace those 2-4 million that are lost. People have started for 15 years to inject stem cells in the heart of various derivations. Cells from the bone marrow, cells from adipose tissues, groups where have been publishing that there are cells in the heart that behave as stem cells, so they can grow, expand, and then differentiate into muscle cells. And all this now has come out to be either fraud or false. So there is no evidence that there are stem cells of any sort that could regenerate the heart, despite thousands of patients being injected from cells. This has been a big scandal. Harvard University has asked for the retraction of more than 30 papers in top journals, and have refunded $10 million to the NIH for fraud of one of his investigators. So basically, there is no hope for stem cells of any kind to regenerate the heart, as there is no evidence that stem cell injected directly as stem cells can regenerate any organ which is not the bone marrow, the skin, or the cornea at this moment. So basically, there is a consensus now in the field. These are 15 top laboratories in the field who signed the consensus statement that basically forget about regenerating the heart with stem cells. You can regenerate the heart using stem cells, but in the laboratory. So you take embryonic stem cells, you obtain 1 billion embryonic stem cells, you convince these cells to become cardiomyocytes, and you inject these muscle cells in the heart. which is a very laborious treatment if you think that the prevalence of heart failure, which in 80% of the cases is due to myocardial infarction, is 23 million people in the world, of whom 80% are in developing countries. So think of having 1 billion stem cells, obtaining 1 billion muscle cells and injected in the heart. Very, very problematic. This is tested in monkeys by Chuck Murray in Seattle, and he's bringing now to clinical trial, but obviously it's not a medicine that could be developed for everybody. However, there are animals that normally regenerate the heart. For example, fish regenerates the heart, amphibians, salamanders regenerate the heart. This is an example of axolotl. This is an amphibian who, the scientific name is Ambystoma mexicanum, lives in the volcanoes of Mexico on 4,000 meters. There was an axolotl colony here in Ljubljana. I remember Radovan taking me in the old, where is Radovan? He's around. Taking me in the old days to see these axolotls close to here to Trieste. Well, you can take these animals, you damage the heart, it regenerates, you damage any organ, it regenerates. You even cut a limb, and in 40 days, the limb completely regenerates. But it regenerates completely in terms of anatomy, physiology, bones, muscle, nerves. So these animals have kept the capacity of regenerating. How do they regenerate these organs? Are there stem cells? Not at all. They regenerate because the already differentiated cells go a little back in their differentiation profile and start proliferating. So it is regeneration of the heart in these animals is proliferation of existing muscle cells. And so a few years ago we thought, well, let's forget this frenzy of stem cells. Let's find a way of convincing existing cells in the heart to again proliferate as they used to do before birth. The problem is obviously how we can do that. And the ICGEB in Trieste, it is more than 15 years that we have developed a facility for the production of a small virus that can be used as a vector. This small virus is called AAV, which stands for adeno-associated virus. Lorena Zentilin, who is there, is heading this facility, and she has produced more than 2,000 AAV preparations. It's a very small virus. It just has a single-stranded DNA as a genome and 60 proteins as the code. And you can engineer this genome to have this virus delivering any gene you want. And if you take this virus and inject in the heart, it will go exactly into cardiomyocytes. So we have a perfect vector to deliver genes to the heart. And when we discovered this, we were immediately happy, and we started delivering genes that might promote cardiac muscle cell proliferation. And we went on the information in the literature on development, which are the genes that during development push cardiac muscle cell to proliferate. We obtained AAV vectors expressing each of these genes and tested either these alone or in combination after myocardial infarction. Complete failure. I mean, educated guess for that didn't bring to anything. Other laboratories did with other genes, complete failure, nothing. However, if you think in biology research, the big discoveries didn't come through educated guesses, but they come from functional screens. For example, this is a picture when one of the reasons why I stayed in Trieste after my PhD is because I had to go to the Stuart Aronson laboratory, the NIH. I was accepted as a postdoc after my PhD, and he was working on oncogenes. And then a fellowship came out to join in 2 years the newly established International Center for Genetic Engineering and Biotechnology, which was nothing at that time. And I took this fellowship, and so I went to do my work in Pavia with Arturo Falaschi. This is how I became associated. But Aronson attracted me a lot because in those years, so in the 80s, late 80s, he made an experiment in which he took cDNAs from cancer cells and put the cDNAs into a monolayer or 3T3 cells. These are fibroblasts that stop proliferating. But then, upon the action of some of these cDNAs, there were foci forming, and he understood what this foci contains, and they expressed a cDNA for the RAS protein. This was the discovery of oncogenes. So it was a functional screen for cDNAs that could impart proliferation. Here at the ICGEB, we have been working with HIV for several years. And you know that it was immediately clear in 1983 when HIV was discovered that the CD4 molecule was a receptor for HIV. So people immediately took HeLa cells, so standard cell line, inserted the cDNA for CD4, and then tried to infect these cells with HIV. Nothing happens. HIV doesn't go into CD4 cells only. And so it was immediately clear there was something else missing. And this something else missing was discovered 13 years later by Ed Berger at the NIH, because he took a library of CD4 cells and transfected this library into HeLa CD4 cells and found one molecule that was the co-receptor assisting CD4 for infection. This was the famous chemokine receptor, CD6-CS4 in his case. But several molecules that are in treatment for tumors, they target enzymes that have been selected by functional screenings. Yamanaka discovered his 4 factors to convert any cell into an embryonic stem cells by testing 21 transcription factors in various combinations and finding those 4 that were acting in this manner. So we reasoned that we need a functional screening. Forget about how it works. We need something that works. And then we go back to see how it works. And so at the ICGEB, we set up a facility, which is a high robotic facility, but at an academic scale. It's not enormously big like you find in pharmaceutical industries, but it is very well equipped because it has a high content microscope and a robot. And it is equipped with libraries with siRNAs against all genomes in the genome, so approximately 18,000 siRNAs for humans and mice. It has microRNAs. It has inhibitor microRNAs, all the FDA-approved drugs. And it has a library of cDNAs coding for 1,200 factors expressed by the genome to be secreted outside the cells. We love this biology of screening. So over the years, we have performed several screenings. If you're interested in screenings of genetic material, so siRNAs, microRNAs, we are happy to collaborate. And we fell in love in particular with the microRNA networks, because you know that our genetic information contains the information to make about 20,000 proteins. These are the textbook genes. But in addition to these, there is information to form other 2,000 small RNAs that are small RNA molecules which are not translated into proteins, but they are progressively matured, they reach the cytoplasm, and then eventually they become molecules of from 21 to 23 nucleotide long, double-stranded RNA, and each of these microRNA targets the mRNA, so the messenger RNA, for tens of hundreds of different genes. So they are big rheostats that lower simultaneous expression of tens of hundreds of genes. And so they have been evolved by evolution, selected by evolution, to target complex biological function, like converting a non-replicating big cardiac muscle cells into a replicating cells. So to make a story short, we screened the library with all human microRNAs, and we ended up, a few years ago, in finding 40 microRNAs that are very effective in converting a cardiomyocytes, that is a non-replicating cells, into a cells that starts replicating, and delivered with an AV vector. These are the 2 most effective, 590, 199A, to an infarcted heart in mice. This is the left ventricle. This is a big scar after 12 days and 60 days in a control. So this huge scar, a human would never live with such a big scar. Well, if you inject the microRNA, they convert this big scar into a much smaller one. This was the first demonstration that you can achieve regeneration in the heart using the capacity of cells to proliferate. Over the years, we learned a lot about these microRNAs. For example, we know that they really work on cells that are cardiomyocytes. So they convince cardiomyocytes to replicate. There are no stem cells here. We know that out of the 40 that we found to work in mice and rats, 8 work, they are all of human origin, work very well in cardiomyocytes from humans. These cardiomyocytes were obtained from embryonic stem cells or for abortion fetuses in collaboration here with the Children's Hospital in Trieste. And we know that they work also on already established big cardiomyocytes. These big cardiomyocytes, in response to these microRNAs, they start proliferating. Here, proliferation is seen by incorporation of an analog of thymidine, so it means that there is DNA synthesis in these cells. And recently, we also learned the mechanism of how these microRNAs work. I will be very short on this because this is very technical. Basically, all of them target a pathway inside the cells which converges in the activation of one transcriptional cofactor which is called YAP1. So this is a very well-established regulator of cardiac cell proliferation during development. So they inhibit a series of inhibitors of this transcriptional cofactor, which is free to go to the nucleus and activate a gene expression program that leads to proliferation of these cells. In doing so, what they do is also convert the cytoskeleton made of actin in the cell into a polymerized form. So proliferating cells have a roundish appearance, and actin polymerization is necessary for proliferation. These are examples of replicating cardiomyocytes. These monstrously big cells that disassemble their cytoskeleton. Here you see everything is fuzzy, there are no more striations, and they start entering the S phase, and here are all the phases of mitosis. Here, for example, there is a non-replicating cardiomyocyte with a beautiful striation, and this is a replicating cardiomyocyte with this fuzzy appearance of the sarcomere. These are pictures of Chiara Collesi, who is our master in taking these fantastic pictures. Obviously, regenerating the heart in a mouse can be relatively easy because the mouse heart is very big, small. But regenerating the heart in a pig is much more difficult. And the heart of a pig is very similar to the heart of a human. So basically, the key experiment was asked is, do these microRNA work also in a large animal? So we teamed up with Fabio Recchia, Giovanni Acquaro, and their team in Pisa. They have a very nice pig facility. They are physiologists and molecular resonance imaging cardiologists. And we started an experiment in which we tried to mimic a human situation. So we had a pig in which we occluded a branch of the coronary artery for 90 minutes. Think of a patient who has a myocardial infarction. Within 90 minutes, he's brought to the hospital, revascularized. So we released the ligature to the coronary artery. And then in the infarct-bordered zone, so in the region that is not dead because of the infarction, we injected in 10 sites an AV vector expressing the most effective of these microRNAs, 199A, which besides being the most effective, was also completely conserved. So it has a sequence which is exactly identical in humans, mice, rats, and pigs. Efficiency of gene transfer was monitored by various molecular means. Where you see black here, this is in situ hybridization. This is where the microRNA goes. So this is the big lesion, and this is where we could inject. The controls far away from the injection. These are slices from the heart. See, this is here there is an infarct-bordered zone. Here the vector was injected. We know that the microRNA works, and the results were truly spectacular. The cardiologist who did the magnetic resonance imaging did this blindly, and he didn't want to believe that after 1 month, the pig that he was looking at was a pig that 1 month before had myocardial infarction. And you see here that at 2 days, MRI showed the same size of infarct. At day 28, those animals who received the microRNA, the infarct was much smaller, and cardiac function was restored almost the same levels as untreated pigs. And at the same time, fibrosis, which is here in blue, the hallmark of what happens after myocardial infarction, was replaced by much more contractile tissue. Showing you just some images of these. These are 5 slices of magnetic resonance imaging. This is the apex of the heart, this is the base of the heart, from the apex to the base. This is the same animal. This is a control animal seen at week 1, week 4, and week 8. This is the left ventricle, big infarct, contrasting in red in the septum, in the anterior wall, which progressively becomes a big scar in 2 months. This is a treated animals, big infarct, same size at week 1, progressively it gets healed. This is a higher magnification, big infarct, progressively disappear. I also have a movie to convince you better. This is a heart 1 month after myocardial infarction. This is the left ventricle, the right ventricle. I make it moving. You see that the heart contracts little. There is a very little contraction inside. This is very thin. This anterior wall is immobile. And these are 2 animals that have been injected with this AV vector. And you see that there is much more muscle mass. And one doesn't need to be a cardiologist to see that contraction is much improved. And this correlated with massive proliferation of cardiac cells in the infraborder zone. Here proliferation is seen in brown with an antibody against this antigen called Ki67. This is the infarct region. And these are all big proliferating cardiomyocytes. Here are other 2 pictures, the infraborder zone, big proliferating cardiomyocytes in the infraborder zone. And these cardiomyocytes go through all phases of the cell cycle. So for example, this is staining for phosphohistone H3. We mark cells that are in the M phase. And you see that these cardiomyocytes have replicated or are going to replicate. There are many of these pictures here. So this is the first time that one can see replication of the heart cells in large animal. There are even mitotic figures, trains of nuclei within the same fibers, and so clear evidence of proliferation. Cardiomyocytes in pigs are heavily nucleated. In humans, there are almost 1 or binucleated. In pigs, there are as many as 8 nuclei in the same cardiomyocyte. This is a control heart, and this is a heart in which the microRNA was injected. This is another sample. You see how many of these cardiomyocytes have undergone replication. So the short-term effects at 1 month were spectacular. These are summarized here. This is, for example, the infarct size in red are the treated animals, in black are the control animals. And these are individual animals from day 2 to day 28 to see the change. You see the infarct size diminishes very significantly, and the function of the heart is the ejection fraction, so how much blood is pumped out every heartbeat is very significantly increased in these animals. The long-term effect, however, were not so beneficial, and several animals died, because if you keep a pro-proliferating microRNA active throughout the life of an animal, because there is no way of stopping expression of this microRNA using viral vectors, the animals, after they stay well, 1 month, 1 month and a week, 1 month and 3 weeks, 7 weeks, they start developing arrhythmias. Several of these animals start dying almost in a synchronous manner. So this microRNA probably induce proliferation. When it is too much, there are arrhythmias and the animals die. They die, they have all implanted a recorder, and so this is a normal ECG, and sometimes they develop a ventricular arrhythmia, and then progressively they die. And the reason why they develop arrhythmia could be related to the presence in several of these animals of a strange number of cells that are proliferating inside the heart. And we characterize these cells. They are not inflammatory cells. They are very small, highly proliferating, so positive for this marker. And they express markers of early myogenic differentiation. So they are a sort of artificial cells that doesn't exist in nature, that resemble a muscle cells at the early stages of differentiation. So they are positive for this marker GATA4 for myogenin, which marks rhabdomyosarcoma, so malignant sarcoma of skeletal muscle, but is never expressed in the heart normally, or in the endothelin receptor that is expressed is most muscle cells. So we believe that this is sort of artificial induction of an early myoblast progenitor. When I was presenting this last November at the meeting of the American Heart Association, which is the largest association in the United States for heart research, they may do meetings with 30,000 people. Somebody from the audience told me, well, you are so enthusiastic of these results, but you are the only person in the world who has induced a tumor in the only organ in the body that doesn't produce normally a tumor, because you know that there are cardiac tumors are very rare. And in fact, I'm very happy because for the first time we have an agent that works. We have a therapeutic gene. The only way we have to find is how to deliver this in a controlled manner. But this is very easy with microRNAs, and we know it already works in mice. because we can use these as a synthetic molecules. So we go to the chemical laboratory, or simply we order them, and we have 23 nucleotide double-stranded RNA corresponding to 199. We inject this microRNA in the heart of mice. It stayed there for 12 days, and it is active because we know it downregulates some specific target genes. And 12 days are sufficient if it is injected in the neonate to convert these ventricle mass into this huge ventricle mass. And you see that these ventricles are much bigger. They are much bigger because they have many more proliferating cells. You can see how many red, or this imagine how many red dots there are in these ventricles. If I do a higher magnification, these are really replicating cardiomyocytes. And if you do the same injection after myocardial infarction in adult mice, the big infarct that you see here becomes much, much smaller. So I believe that an idea by which this patient is brought to the interventional cardiology unit is reperfused by angioplasty. And at the end of reperfusion, or 1 or 2 days later, a cardiologist again insert a catheter in the coronary artery and inject a solution containing the synthetic microRNA. The synthetic microRNA enter into the border zone of the infarct and push the survived cells to proliferate to heal the myocardium might be feasible. And this is what we would like to attempt. There is an IP, a patent from the ICGEB, and obviously needless to say that pushing this to clinical application would be a huge benefit, as you can imagine, seeing the breadth of the problem also for the ICGEB community. In conclusion, cardiac regeneration can be achieved through the stimulation of endogenous cardiomyocyte proliferation, but this process needs to be tightly dosed. I take once more the opportunity of thanking the many people who contributed to this work, and I take pride saying that this work was all conducted here in Trieste with the collaboration of the Pig Stories and Fabio Recchi and his team in Pisa. You have seen the names of the people who contributed to this work. The main actor, Hashim Ali, who is an expert in proliferation. Chiara Collegio, I already mentioned her. Luca Braga, he runs our high-throughput screening facility. Consuelo Torini discovered the mechanism of action. Giulia Prosdocimo is the first author of this big paper that was published last week. Ilaria Secco is also contributing heavily to the study of cardiomyocyte proliferation. Lorena Zentilina, I also mentioned her later, before she runs our AV facility, and we collaborate here with the clinical group cardiologists led by Gianfranco Sinagra. Serena Zacchigna has accompanied me throughout these last, how many years? A bit less than 20 years in all this interesting intellectual travel throughout all the projects we have carried out, and she now, since a few years, runs an independent group here in Trieste. And I thank you very, very much for your attention.